Okay. So, can't see. There we go. Okay. So here, as I kind of transition over into what in the world are we supposed to be looking at here, here is some mitosis slides. Okay, this is over here on this side, this is an onion cell. Over here on this side, this is a skin, cheek scraping. Okay. Pay attention to the animal. Okay. Pay attention to the animal cell. I'm not too concerned about you being able to diagnose what a cell looks like in a cucumber. As delicious as they are, I'm not worried about that in a human anatomy class. Right? But this is interface here. And even though you can see a number of things going on this slide, I'll mark it. This cell right here. Okay. This cell right here. Okay. When you look at the nucleus, the defining characteristic of interphase is that the nuclear membrane is intact. And when you look really closely out there, and if I get really close to my screen, okay, you can see this is the nucleus. Okay, that's the nucleus. And you can see around that sucker, you can see a nice little line there. That tells you that you're looking at interphase. Same thing down here in this one. There's the dark line around the nucleus. That's the nuclear membrane. It is intact. Okay. If I look, because it says also there's prophase, so let's look around. Let me find a good one anyway. Here, I don't see a nice dark rigid line. Here, I don't see a dark rigid line around the nucleus like I do here, like I do here. Okay, the two dotted ones have a dark line around the nucleus. Okay, so an intact nuclear membrane. Here, it looks like there's a portion of it here, but then over here, it looks like a certain fragment. Here, I don't see it at all. This tells me here that I'm looking at prophase, so that's prophase. There's an example of prophase. That's an example of interphase. That's an example of interphase. Okay, it's that nuclear membrane that gives it away. This is the easiest one. Okay, it's right here. Okay, it's right here. In metaphase, the chromosomes meet in the middle of the cell. Doesn't get any easier than that. Hell, it's even easy on an onion slide, right there. In anaphase, those chromosomes pull apart. Okay. The second easiest one to see, right there. Okay. Here I see two dark lines, being one pole to this pole, being one pole to this pole. Here's metaphase, right? Here's metaphase. Here's metaphase. Okay. And now you can see the difference. There's the chromosomes meeting in the middle. Chromosome looks like that. Okay. You get one going this way, and you get one sister chromatid going that way. Okay. Not the best. She could have done a lot better. Okay. With telophase, one of the big things you'll see with telophase, guys, is you'll see one cell have this cleavage furrow in it. There's your cleavage furrow. Okay. That is the hallmark that you will see. And by hallmark, I mean it's something that is always associated with it. Okay? So these hallmarks I've been talking about, meeting in the middle, two dark lines being pulled towards the opposite poles. Okay? Nuclear membrane, no nuclear membrane, cleavage furrow. Those are things that you need to be able to identify in your head to tell you that you are seeing that phase. She has a late telophase, and that's late as hell. This one's a little bit better up here because here's that invagination and there's the slide. Okay, and so here is that cleavage furrow that I was talking about. Okay, so that's telophase. Early, late, I could care two bits about. All right, but that's what you should be looking for. And she made some practice slides for you. Evidently, everything is found there. Sorry. Okay. And Anna, Meta, Tilo, Pro, and Interface. And they're almost all there. I'll show you one more thing, hopefully. Any questions while I'm looking for this thing real quick? Lecture, lab.
Are we going to finish 5.5 organs?
Yeah. Finish it on.
Oh, okay.
Yep.
Oh, going to finish on 2. But the last part of chapter 4, we're responsible to go listen to the video.
What chapter is this? Yes. I mean, and I'll be more than happy to review it on our review day as well for you. Here's a good little telophase right here, and I just use this thing called the Google, and there you go. There you can see that invagination right there. Okay, here you can see that invagination right here. In telophase is the exact opposite of prophase. So everything that we lost in prophase comes back in this phase, but at 40x, you are not going to see any of that nonsense. So it almost looks like anaphase. The difference between anaphase and telophase for us at 40x is the presence of this cleavage furrow. Okay, here's that cleavage furrow right here. Cleavage furrow. Cleavage furrow. All right. Pole to pole. So this is one cell that is becoming 2. So what will we do? What will we do on Tuesday? Tuesday, where am I at? Here I am. So Tuesday, we will finish up the last kind of, what do we got? 1, 2, 3, 4, 5, 6, 7. There's like 8 slides left. We can lecture and finish them up if you want, or we can finish them up on Tuesday. I can go over the cell cycle. I will go over the cell cycle for those of you who would like me to go over that, I can do that. And any questions that you have over the material, I'll give you a breakdown for the exam, because that is what? That's 9-3. All right. What is this? Day 27th? Yeah.
So the slides you were showing with the phases of cell division, the first ones, that's from the lab. Those slides, we can find them
Yes.
in the Connect lab
If you go to, wrong one.
or the course.
If you go to Blackboard, coursework, click on labs, you have pictures of tissues, you have histology, you have mitosis slides, and that's where I got those mitosis slides from.
Okay, thank you.
If I show you, let me show you this. No, let me pull that up. All right. Here is your A&P revealed. Now you have your my course content. I haven't entered the code in yet, so I haven't pulled it up. But here again, I clicked on this one the other day, and here is your highlighted. There is your simple squamous epithelium, just like I pointed out the other day. At the bottom is my simple cuboidal, okay? Cube shaped. I'm curious. Yep, simple cuboidal. Look towards the free edge. Here's the lumen of the tube. This is the center of this tube. This is your, I believe this is the kidney tubule slide that I was mentioning earlier in lecture. Okay, but this is the center of the tube, and here's the free edge. Okay, here's the center of the tube. There's the free edge. Okay, there's the free edge. There's the tube. These are all simple cuboidal epithelium. If I go in, there you go. Simple cuboidal epithelium. If I was to use this on the exam, identify the highlighted tissue. This is ciliated simple columnar. You don't need to give me ciliated, but this is ciliated. You can see that little fuzzy little border. These are very tall. Okay. This is an undulating type of tissue here. Okay. So that's why the nuclei appear at weird, funky levels here, but this is simple cuboidal, or I'm sorry, simple columnar epithelium. All right, and if I highlight it, there you go. Okay. There's the columnar cell individually. Non-ciliated, there you go. There's those goblet cells that I was talking about. You see those little white pockets? Okay, right there. And there's your tall, and there they all lined up in a row. All lined up in a row.
Where's the base in that? That's really difficult to tell.
Does it give me highlighted for your pleasure?
Oh.
There you go. This part here looks cellular in structure, but that's some other layers. Okay. There you go. It's going to contain blood vessels and all sorts of other stuff, and we'll get into that later when we get into the digestive system. Here's pseudo. Okay. Here you can see, right? They're all at various different levels. Okay, so pseudo-stratified. Okay. It's ciliated, I just clicked on it to get rid of the highlight. So again, it's those nuclei. Very easy to identify. And you can see if they're all lined up in a row. To that end, if I get, let's go here, okay? There's stratified columnar. All the nuclei. All the nuclei. All nice and neat lined up. This looks almost cube shaped, but this top one is column shaped. Go back one, whoops, wrong button. Cubed. And for the most part, this is cubed, although it almost appears columnar, at least to me at first glance. Okay. But this is stratified cuboidal. How do I know? Look at the nucleus. If this was columnar, this would be a little bit more taller because that nucleus is always at the bottom third. What's up, buddy?
Hi.
Here's stratified squamous epithelium. All of this is stratified squamous epithelium, okay? From this bottommost portion all the way to the top, okay? Then I have areolar connective tissue underneath that, right here, okay? And then down a little further in the dermis, I have some dense irregular connective tissue. Here's what keratinized skin looks like. Keratinized stratified squamous epithelium. Again, squamous epithelium, stratified squamous epithelium all the way from down here, all the way up here. And all this is your keratinized cells here. Okay, that's the stuff that you slough off, the very top of your skin. Here is loose in nature. And here down here, you can see there's your dense irregular connective tissue. And even down here, you can see this right here. You can see some fat. There's some adipose tissue right there. So on this one slide, I see 3 different tissues right off, or 1, 2, 3, 4 different tissues easily right off the bat. There's a couple others as well, but I don't want your mind to explode just yet. I'm gonna wait till next week, okay?
Would we be able to see that larger? Or
On skin, I'm going to imagine it's blowed up a little further too. I just haven't gotten down there yet.
okay.
So I'm hoping it is. I went through this over the last like 3 days here, so that's fine. Gabby, spell your whole name out for me, please. This is loose connective tissue or areolar connective tissue. There's your collagen fibers, really thick, elastic fibers, thin, fibroblasts. Can of corn, adipose. And reticular, okay? Now, what do you think this can kind of look like? What do you think this gets mistaken for? Anybody?
Dense connective tissue?
This is dense regular connective tissue, but what else do you think this can be confused for? What does this look like?
Cartilage?
Nope.
Muscle tissue?
Maybe muscle tissue, right? This looks like skeletal muscle tissue, okay? They all go in the same direction, but the one thing that this lacks, it does not have alternating light and dark bands that are perpendicular, okay? They give you that perpendicular appearance to it, all right? So this is dense regular connective tissue. If I look, I'm gonna look at this real quick. Here's dense irregular close up. This is what was in that skin slide. Okay. There it is. If I go, while I'm thinking of it here, let me go back to dense regular. Okay, dense regular. Okay, collagen fibers going all in a row. Okay, now do you see the difference here? Okay, what if I have this? What if I have this slide and this does a good job of showing it? I don't know what magnification this is. I don't know if it says. But what if I turn that slide so it's coming like this? And those lines, those alternating light and dark bands are going in the same direction as the dense regular. On an exam, those get confused well, not all the time, because there's always 2 or 3 people that always get it correct. But a lot of people really screw those two up. They flip-flop them, okay? And here, you can really see this alternating light and dark band. So that's what you got to really make sure you see, that alternating light and dark band, because I don't see that I don't see that here. There's elastic, just a buttload of various different elastic fibers. It has its own look. It doesn't look like anything else. No one confuses that one. Here is hyaline. Okay, there's that perichondrium. Okay, there's that thick perichondrium there. Chondrocytes scattered throughout a lot of cartilaginous matrix. Compare that with elastic fibers, and there's your perichondrium. Okay. Now we have a greater number of fibers, elastic fibers, and a greater number of cells. So that's how you can differentiate between these two. I go back to fibrocartilage. I mean, fibrocartilage, you can see those collagen fibers and those chondrocytes. I mean, this is a definitive hallmark view of fibrocartilage. Okay, fibrocartilage looks drastically different from hyaline. You don't really see the fibers. This one you do. This one you do. But there's a buttload more in elastic. That's a technical term. Oops, wrong button. Compact bone. I mean, you can honestly hold this slide up to the light and identify everything on this one. This one's really easy. There's your osteon. Whoops, there's your osteon. There's your osteon. I think I can, there you go. I don't even have to do it, okay? Pretty easy. That one's can of corn. Blood, can of corn. Red blood cell, platelet, leukocyte, erythrocyte, platelet, white blood cell. Pretty easy. Those two are the easiest. Again, they're skeletal, alternating light and dark bands. Now, here, you can see it right here, okay? I'm focusing in right through here. Here's this fiber coming in. You can see my little arrow there. Here it also comes up and it branches, it bifurcates. And you see that phenomena right here. Here's the fiber coming in and it branches. You can also see those intercalated discs right there. I think I can highlight those. Yep. There's the branching that I was talking about. There's those intercalated discs. Okay, there's all the nuclei. I just clicked on nuclei to click off this intercalated disc. So again, alternating light and dark bands. I can see those alternating light and dark bands right there. Okay, I can see it in all of it, to be honest, but I'm right there because it's very easy to see right there in the center. But what do these things have? Once I see alternating light and dark bands, connective tissue, I'm in the middle. A lot of fibers. Yeah, there's a lot of fibers here, but I see some striations, and I see striations only in muscle. So here, I have striations, muscle. Okay, now is this skeletal or is it cardiac? Because those are the only two. And I see those dark perpendicular lines there, so boom, cardiac muscle. I don't know why. Nervous tissue, can of corn as well. There's the neuron, axon, and a bunch of little dendrites coming into it. That one's super easy. Now, why did this one not work? Oh, we have different views. Low magnification, high magnification. Here you can see that spindle-shaped appearance to the nucleus. That's the spindle-shaped, and if I just look at the outline, okay, there's the outline, there's the spindle-shaped kind of appearance to that cell. Spindle-shaped appearance to the cell, okay? So it gives that undulating appearance. That's with high magnification. You're not gonna see it. You don't see it that well with low magnification. I mean, I can because I've seen this a billion times. So, I mean, I can see the outline of that individual cell right there. I can see the ends that are tapered on the nuclei here that I can see. So for me, that's enough for me to say that this is smooth muscle. But for you guys, I'm going to put it probably on this one so you can see it a little bit better. Okay? Because I'm going to assume this is the first time most of you are seeing tissues. You don't really sit around and look on Instagram at tissue slides, I'm going to imagine.
For the lecture exam, do we have to identify the different kinds of tissues from these pictures, or is that just a lab exam thing?
I generally just try to keep that to lab.
Okay, so on the lecture exam, it's more like words, just identifying,
Words,
describing.
Yep, yep.
Okay.
Does that help you guys a little bit?
The lab exam will also have cell cycle on it, though, the things you just went over in the very beginning. The cell division.
Mitosis, epithelial tissue, connective tissue, skin, and then skull. It's also the skeleton as well. Skull, axial, appendicular skeleton. We haven't gotten to that yet, so I don't imagine we're going to until after maybe a little bit on. Next week we'll introduce some stuff. So Tuesday, we'll finish up the notes. There's only 8 slides. We'll finish it up. We will also finish up, we'll review mitosis, okay? That won't be too much. That won't be too difficult of a concept for you guys. And then we will review. And by review, I mean, do you have questions? Concepts that you have some questions over? Ask them, okay? I'll give you the breakdown for the exam as well on Tuesday.